Hi all, welcome to Gallant IAS. Today we are discussing about the topic phenomenon of infection and infection process. Okay. Phenomenon of infection and infection process. Okay. So this topic which comes under the plant pathology part. Okay. So this plant pathology, which is the first unit of paper 1. Okay. Have a look on to the previous year questions from this part, phenomenon of infection and infection process. So the first question is, explain the process of infection by pathogenic fungi. Okay, 50 mark question which is asked in 2019. Okay, the question is process of infection by pathogenic fungi. Okay, the next question is, explain the molecular basis of infection and disease resistant in plants. Okay, this is a 20 mark question which is asked in 2016. Okay, here the second question which having two parts. Okay, the first part is demanding you to write about the molecular basis of infection, and second part is the disease resistance. Second part is the disease resistant in plants. Okay, so you need to address both the part of the question. We have gone through the question. Okay, so the topic we are discussing is the infection process and phenomenon of infection. Okay, so this topic which comes in unit 1 of paper 1 under plant pathology. Okay, so before moving on to the topic, we will discuss about the major contents we are discussing under this class. Okay, so firstly we will look on to the general terms with regard to pathology. Okay, so after discussing the general terms, we are moved on to the infection process. Okay, we move on to the infection process. Okay, we will discuss what infection process is, and after that we will discuss about the factors which plays in the infection process. Okay, the first is the host factor. Host factor will have a role there, and second is the pathogen factor. We will discuss all in detail what are host factors, pathogen factors, and the third one is the environmental factors. Okay, so under infection process we will discuss about the host factors, pathogen factors, and environmental factors, and then we move to the infection, and under infection part mainly we are discussing about three major topics are there. First is the pre-penetration, penetration and post penetration. Okay, so infection means it leads to a series of events. First is the pre-penetration events, and next comes the penetration, and the last one is the post penetration, and the last is we discuss about the exit of the pathogen. Okay, the last topic we are discussing is the exit of the pathogen. Okay, so these are the major topics we are discussing under this class: infection process and phenomenon of infection. Firstly, we will discuss about the terms in pathology. Okay. The first term we are discussing is the disease. Okay. So we all know what is a disease. Okay. So any malfunctioning of host cells and tissues that result from the continuous irritation by a pathogenic agent or due to some environmental factors and lead to development of symptoms. Okay. So simply disease means it's a malfunction as a action of the pathogenic agent. The action of a pathogenic agent, it causes malfunctioning of host cells and tissues. It causes malfunctioning of host cells and tissues. And that malfunctioning of host cells and tissues which lead to the development of symptoms, and that is simply what we called as disease. Okay. So environmental factors also plays a role in the disease process. Okay. Environmental factors having a role in this disease process. Okay. So disease means malfunctioning of host cells or tissues by the action of a pathogenic agent, and it leads to the development of symptoms. Okay. So the action of the pathogenic agent which leads to a development of symptoms, then it is called as disease. Okay. Next is pathogen. Pathogen means an entity. Okay. An entity which can usually a microorganism that can cause disease on a particular plant. Okay, that is what we called as pathogen. Okay, so pathogen is an entity, usually a microorganism that incite disease. Okay, in a literal sense, a pathogen is any agent that cause damage to the host cells or tissues. Okay, in literal sense, we can define that pathogen is any agent. Pathogen is any agent that cause damage to the host cells or tissues, and it causes damage to the host cells or tissues. Simply it can termed as pathogen. Parasite, organisms which derive the materials. Okay, parasite means they mainly depend on other organisms. Okay, parasite means they mainly depend on other organisms. Okay, so organisms which derive the materials. Okay, so here the materials means the materials they needed for their growth and development. Okay, so these are parasites. So they depend on other organisms for their food and nutrition. Okay, so organisms which derive the materials they need for the growth from living plants or host. Okay, that are what we called as parasites. And next is the pathogenicity. Okay, so what do you mean by pathogenicity? Pathogenicity means it is the ability of pathogen to cause a disease. Okay, pathogenicity means it is the ability of a pathogen to cause a disease is called as pathogenicity. And pathogenesis. Okay, pathogenesis means the chain of events. Okay, pathogenesis means it's a chain of events from the initial contact to the symptom expression. Okay, that entire process what we called as the pathogenesis. Okay, so it's a series of event or chain of events. Okay, pathogenesis means it's a series of event or chain of events from initial contact to symptom expression. Okay, so to make the symptoms on the plants, it must make a contact with the pathogen must make a contact with the host plant. Okay, so pathogenesis means the entire events from the initial contact to the symptom expression. This entire process what we called as the pathogenesis. And next is sign. Okay, sign means the pathogen or its parts or products seen on the host plant. Okay, sign means the pathogen or its parts or products seen on a host plant. Okay, so here the product means the products of the pathogen. Okay, either the pathogen or its parts or its products which are seen on host plant. Okay, that what we call as the signs or the parts or the products of pathogen which are seen on the host plant, what we called as the sign. Okay, symptom means it is the external and internal reaction. Okay, so symptoms means it is the external and internal reaction or the alterations of a plant as a result of the activity of a pathogen. Okay, symptom means the external or internal reactions or the alteration of a plant by the action of a particular pathogen. That is what we called as symptom. Okay, so normally a pathogen which may act on the host plant by the action of that by the action of that particular pathogen, it may cause disease on the host plant. Okay, so symptom means the external, both the external and the internal reactions or alterations of a plant as a result of disease. Okay, and the last one which is syndrome. Okay, syndrome means the set of varying symptoms. Okay, so, syndrome means the set of varying symptoms characterizing a disease are collectively called as syndrome. Infection process. Infection process means establishment of pathogen in the host plant. Okay, so infection process means it is the establishment of pathogen in the host plant. Okay, once they may come in contact with the host plant, they entering into the host plant, and then once they enter into the host plant, they establish inside the host plant. Okay, so entry and colonization of pathogen in the host tissues is known as establishment. Okay, entry and colonization. Okay, so once they may come in contact with the host plant, this pathogen may enter into the host plant. Okay, first is the entry. Okay, so once they enter into the host plant, what happens? It multiply or grow in the host plant. Okay, so once they enter, they multiply. Okay, or they colonize inside the host plant. Okay, entry and colonization of pathogen in the host tissues is known as establishment, and infective propagules coming in contact with the host are known as inoculum. Okay, and the infective propagules, okay, the entry of the pathogen into the host tissues is known as establishment, and infective propagules coming in contact with the host are known as inoculum. Okay, the success of process of infection depends upon three factors. Okay, the success of this infection process is mainly depend upon three factors, and the factors are the host factors, pathogen factors, and environmental factors. Okay, so which are the factors? Host factors, pathogen factors, and environmental factors. We will discuss all in detail. First is the host factor. Okay, host factor. The first we are discussing under the host factors are the susceptibility of host. Okay, so it is genetically controlled by DNA. Okay, so the susceptibility of host which is genetically controlled by DNA, and it is an inheritable character. Okay, so what do you mean by an inheritable character? It is transferred from the parents to the offspring. Okay. So the susceptibility of the host which is controlled, genetically controlled by DNA, and it is inheritable. That means it is transferred from the parents to offsprings. Okay. Next is the disease proneness of the host. Next is the disease proneness of the host. It is decided by external factors. Okay. So disease proneness of the host means it is decided by the external factors such as host nutrition. Okay. So here disease proneness of the host. Okay. That disease proneness of the host which is decided by external factors such as host nutrition. Okay. For example, if more nitrogen application makes the host more susceptible and more potash application lead to less susceptibility. Okay. So here the proneness of the disease proneness of the host which is mainly decided by the external factors such as host nutrition. Okay. So if nitrogen application make the host more susceptible and the potash application leads to less susceptibility of the host. Next is the pathogen factor. Okay. Pathogen factors means virulence and aggressiveness of pathogen. Okay. It is determined by genetic material which is inheritable character which is transmitted from parents to offspring. Okay. So virulence or aggressiveness of the pathogen. Okay. What do you mean by virulence? The ability to cause disease. Okay. That is what we called as ability to cause disease. Okay. That is what we call as virulence. Okay. So virulence or aggressiveness of the pathogen, it is determined by genetic material which is a inheritable character which is transmitted from parents to offsprings. High multiplication rate of the pathogen. Okay. The other factor which is the high multiplication rate of pathogen. Okay. With common logic we can guess that. Okay. Chances of infection. Okay. So if the pathogen which having high multiplication rate, chances of infection increases with a high rate of multiplication. Okay. So here the pathogen which having high rate of multiplication means the chances of infection increases. Okay. And proper inoculum potential. Okay. In case of specialized pathogens, very few or even one spore is capable of causing infection successfully. Okay. Next is the proper inoculum potential. Next is mainly dependent. We have discussed inoculum, right? Okay. So next is the inoculum, proper inoculum potential. Okay. Proper inoculum potential. Okay. So in case of specialized pathogens, a very few or even one spore is capable of causing infection successfully. Okay. For example, consider this as a spore, right? Okay, there is only one spore is there. Okay. So here this is a specialized pathogen, and this single spore which is capable of creating infection in host plant. Okay. So specialized pathogen means a very few or even one spore is capable of causing infection successfully. Okay. That is the inoculum potential. Okay. So that one spore is enough to bring up the infection successfully. Okay. Whereas non-specialized pathogens require high density of inoculum. Okay. On the other hand, consider that non-specialized pathogens, they require high density of inoculum. Okay. So this high density of this pathogen is capable of creating disease on the particular host plant. On the other hand, the specialized pathogens which are very few in number or even a single spore which are capable of causing infection successfully. The third factor, which is the environmental factor. Environmental conditions such as temperature. Okay, environmental factors means the environmental conditions such as temperature, relative humidity, moisture, etc. are very important for survival, dissemination and infection process. Okay, so environmental factors such as temperature, relative humidity, moisture, etc. plays a very important for the survival and dissemination and infection process. Okay, process of infection can be grouped into 3 stages. Okay, the infection process which can be grouped into 3 stages. Okay, so which are the 3 stages of infection process? The first one is pre-penetration, second is penetration, and the third one is post-penetration. Okay, so the infection process can be grouped into 3 stages. First is the pre-penetration, second is penetration, and third one is post-penetration. Pre-penetration. Okay, depending upon plant pathogen activity, the plant pathogens are classified into 2 categories. Okay, in pre-penetration, depending upon the plant pathogen activity, the plant pathogens are classified into 2 categories. Okay, which are the 2 categories? One is active invaders and the other one is passive invaders. Okay, so depending, it is mainly depending, this classification which is mainly depending upon the plant pathogen activity. Okay, this classification is mainly by the plant and pathogen activity. Okay, so it can be grouped into 2. One is active invaders. One is active invaders and other one is passive invaders. Active invaders and passive invaders. Pathogen which makes an aggressive effort to gain entry into the host cell. Okay, so active invaders means the pathogen or the infection agent which causes an aggressive effort. Okay, they makes an aggressive effort to make an entry into the host cell or tissues. Okay, that is what we called as active invaders. Okay, and the other thing is that this active invaders does not require any external agents to gain entry into the host cells. Okay, so what do you mean by active invaders? Active invaders means pathogens which make an aggressive effort. Okay, why they make an aggressive effort? To make an entry into the host cell or tissues. Okay, they make an aggressive effort to make an entry into the host cell. That is what we called as active invaders, and they do not require any external agents to gain entry into the cells. Okay, so examples of active invaders are phytopathogenic fungi and phanerogamic parasites. Okay, these phanerogamic parasites and phytopathogenic fungi both are active invaders. Okay, and on the other hand, passive invaders means no aggressive effort. They does not show an aggressive effort to make an entry into the host cell or tissues. Okay, so passive invaders does not show an aggressive effort to make an entry into the host cell or tissues. And this passive invaders which requires the help of external agents. Okay, so here the external agents means like insect vectors or wounds caused by agriculture implements. Okay, so this passive invaders does not make an aggressive effort to gain an entry into the host cell. Okay, so they they may enter into the host cell by or with the help of external agents. Okay. So, the external agent means the insect vectors or the wounds which are happen on the plant by agriculture implements. Okay. So, the examples for this passive invaders are plant viruses and phytopathogenic bacteria. Okay. The examples for passive invaders are plant viruses and phytopathogenic bacteria. Okay. This viruses and bacteria infect the plant mainly by the wound happen on the host plant. Through that wound, they gain an entry into the host cells. Okay. On the other hand, active invaders means they make an aggressive effort to gain an entry into the host cell, and the examples are phytopathogenic fungi and phanerogamic parasites. And passive invaders examples are plant viruses and phytopathogenic bacteria. Okay, what do you mean by phytopathogenic bacteria? Means the bacteria which are able to cause infection in plants. On the other hand, here phytopathogenic fungi means the fungi which are capable of producing infection in plants. Okay, plant viruses. Okay, plant viruses which are passive invaders. We have discussed that, right? Are particulate in nature and do not have any capacity to enter the host cell. Okay, so plant viruses are particulate in nature. Okay, so we know the property of virus. Okay, this virus which is acellular in nature, they do not have any cellular components. Okay, they are acellular in nature. Okay, so viruses are particulate in nature and they do not have any capacity to enter the host cell. Okay, so they do not make an aggressive effort for entry. Okay, so this plant viruses do not have any capacity to enter the host cell. So they do not make any aggressive effort for entry, but depend on different insect vectors for the entry into the host cell. Okay, so viruses which are capable of infecting plants. Okay, so here we are discussing that these plant viruses do not have any capacity to enter the host cell. Okay, mainly they enter into the host cell through a vector. Okay, vector can be an insect or through the wounds happened on the plant. Through that wounds also these viruses can enter into the host cell. Okay, bacteria have no dormant structures. Okay, in the case of bacteria do not have any dormant structures, hence no pre-penetration activity except for multiplication in infection drops on natural openings. Okay, so bacteria have no dormant structures, hence no pre-penetration activity. Okay, however, nematodes show some orientation towards root surface before actual penetration. Okay, however, nematodes which show some orientation towards root surface before actual penetration. Okay, in the cases of viruses, plant viruses and bacteria, there is no pre-penetration activity, and in case of nematodes there is an orientation towards the root surface before actual penetration. In fungal pathogens, pre-penetration includes spore germination and growth of resulting germ tube on the surface of host plant. Okay, in fungal pathogens, pre-penetration include spore germination and growth of resulting germ tube on the surface of host plant. Okay, in fungal pathogens, pre-penetration include spore germination. Okay, if we are considering this as a spore, okay, this is the host plant surface, considering this as the host plant surface. So here this is the spore, this spore germinate and as a result of germ tube is being produced there, and through the germ tube, it can enter into the host plant. Okay, germination is essential, that change from low metabolic rate to high metabolic rate. Okay, germination. During germination, there is high metabolic rate. Okay, before germination, it is in dormant period or dormancy period. So at the dormant period, the metabolic rate will be comparatively low. Okay, and during germination, there is a shift from low metabolic rate to high metabolic rate. Okay, germination is essentially the change from low metabolic rate to high metabolic rate and involves a change from near dormant to intense active. Okay, so before germination it is in dormant period, that means on dormant period the activity is comparatively very less, and during the germination it shows intense activity for high metabolic rate. Okay, so to high intense activity it requires energy source. Okay, that is as a energy means here it used carbohydrate or fat reserves in the propagules. Okay, to gain the energy or for the germination it requires energy. So high metabolic rate is there, so it requires energy. Okay, for the energy it uses the energy sources such as carbohydrates or fat reserves which are present on the propagules. Fungal inversion is chiefly by germ tubes or structure derived from them. Okay, fungal inversion is chiefly by germ tube or structure derived from them. Okay, fungal inversion. So now we are discussing how the fungal inversion takes place. Okay, so fungal inversion is chiefly by germ tubes or structure derived from the germ tubes. Okay, in some fungi like Rhizotonia zolani and Armillaria melia, the hyphae act in a concerned way to achieve the penetration. Okay, we will discuss how the hyphae act to achieve penetration into the host. Okay. In Rhizotonia solani, the fungus on coming in contact with the root surface first form infection cushions. Okay. In case of Rhizotonia solani, the fungus on coming in contact with the root surface first infection cushion and apozoria, and from this multiple infection takes place by means of infection peg. Okay, so in Rhizotonia solani, the fungus on coming in contact with the root surface. Okay, so here this is a spore. This spore will produce germ tube, and this germ tube may come in contact with the root surfaces, first form infection cushions and apozoria. Okay, what do you mean by apozoria? Apozoria means a swelling at the tip of germ tube. Okay, so here this is the germ tube, and here at the tip of the germ tube there is a swelling happens on the tip of the germ tube. That swelling is what we called as apozoria. Okay, and through a swelling of the tip of germ tube from which penetration peg emerges that serves to pierce the epidermal cell wall or to enter the epidermis to stomatal aperture. Okay, so look on to the figure. Here it is a stomatal aperture, this germ tube which produce or aposorium, or aposorium means it is the swelling at the tip of a germ tube. Okay, and the swelling of the tip of the germ tube which forms the penetration peg. Okay, so you can see that on the figure. Look on to the figure, you can see that on the figure. You can see that the penetration peg which is piercing into the epidermal cells. Okay, so this penetration peg which is piercing into the epidermal cell wall or enter the epidermis through stomatal aperture. Okay, or through stomatal aperture. On the stomata there is stomata means it's a natural opening. Okay, so either this penetration peg enter into the epidermis through the stomatal aperture or by producing the infection penetration peg, it pierces into the epidermal cell wall and then enters into the host cell. Okay, and in case on the other hand, it can enter the epidermis through stomatal aperture. Okay, hostoria means it's a specialized structure which mainly seen in fungi, and this hostoria, it's absorptive in function, it absorbs nutrients and water for the growth, and this hostoria will absorb nutrients and water for the growth and development of fungi. Okay, and in Armillaria milia, the fungus hyphae form rhizomorph. Okay, look on to this figure. So this figure you can see that in Armillaria milia it formed the rhizomorph. Okay, what do you mean by rhizomorph? The fungus hyphae forms the rhizomorph. Okay, so rhizomorph means it's the aggregation of hyphae into robe-like strands. Okay, so in Armillaria milia, the fungus hyphae form the rhizomorph means the aggregation of hyphae into robe-like structure. Okay, here you can see that the robe-like structure, this is the aggregation of hyphae into robe-like structure. It is the aggregation of hyphae into robe-like structures, and through this rhizomorph or this robe-like structures, only through this robe-like structures they can cause infection. Okay, so rhizomorph means it is the aggregation of hyphae which forms a robe-like structure on the surface of the host plant, and through that rhizomorph they can penetrate into the host cell. Only through that rhizomorph they can penetrate into the host cell. Pathogen penetrate plant surfaces by direct penetration or indirectly through wounds or natural openings. Okay, pathogens penetrate plant surfaces. Okay, so pathogens penetrate plant surfaces by direct penetration or indirectly through wounds or natural openings. Okay, so mostly in cases of plant viruses, these plant viruses can enter into the host through the wounds. Okay, we already discussed that, right? So these do not have any specialized structure. These plant viruses do not have any particulate in nature, so they do not have any structures to make an entry or into the host plant or to the host cells. They do not show an aggression towards the host cells. Okay, so they may enter into the host cell through the wounds happen on the host plant. Okay, bacteria enter plant mostly through wounds and less frequently through natural openings. Okay, so we have discussed this, right, regarding the plant viruses and bacteria. We have discussed that there is no pre-penetration in bacteria and plant viruses. Okay, bacteria enter plants mostly through wounds and less frequently through natural openings. Viruses, viroids, molecules. Okay, what do you mean by molecules? The other term for mycoplasma. Okay, so under the microbiology part, we have discussed the structure and multiplication of viruses. We have discussed the structure and multiplication of viroids. We have discussed the structure and multiplication of mycoplasma. Okay, so viruses, viroids, molecules, fastidious bacteria. Okay, so they enter, this fastidious bacteria, viruses, virions, molecules enter through wounds made by vectors. Okay, what do you mean by fastidious bacteria? This fastidious bacteria means they require special conditions. Okay, fastidious bacteria means they require special condition and substances for their, they require special conditions, they require special conditions and substances, they require special condition and substances for their growth are defined as fastidious bacteria. Okay, so fastidious bacteria means they require special conditions and substances. If the special conditions and the special substances is formed, then only the bacterial growth will take place. Otherwise, this bacteria cannot grow further. Okay, fastidious bacteria means it require special substances and conditions for their growth and development. Fungi, nematodes and parasitic higher plant enter through direct penetration and less frequently through natural opening and wounds. Okay, fungi, nematode and parasitic higher plants enter through direct penetration. Okay, fungus, nematodes and parasitic higher plants which may enter into the host through direct penetration and less frequently through natural opening and wounds. Indirect penetration. Okay, direct and indirect penetration is there. Indirect penetration means through wounds. This microorganisms or this pathogen can enter into the host. Okay, wounds. Wounds caused by farm operations, hailstorms or insect punctures etc. will help in the entry of different plant pathogens into the host cells. Wounds happen on the host plant. Okay, if a wound happens on the host plant, through that wound they can enter into the host plant. As we all know that the plant which is having a rigid cell wall and it is made up of cellulose. Okay, so these microorganisms cannot enter into the inside of the cell through this rigid cell wall. Okay, so if there a damage happens to the cell wall, through that damage or through that wound, these microorganisms or these pathogens can enter into the host. Okay, so wounds caused by farm operations, hailstorms or insect punctures etc. will help in the entry of different plant pathogens in host cells. Okay, weak parasites enter through wounds caused by hailstorm and freezing. Okay, weak parasites enter through wounds caused by hailstorms and freezing. Example Macrophomenia fasciolina. Okay, pathogen causing brown rot of fruit. Pathogen causing brown rot of fruit. Okay, so here the infectious agent or the causal organism which is Sclerotinia fruticola enter through wounds caused by insect punctures. Okay, so pathogen causing brown rot of fruits, Sclerotinia fruticola enters through the wounds caused by insect punctures. Similarly, causal organism of Dutch arm disease. Okay, causal organism of Dutch arm disease, Ceratostomela almi enters through the wounds caused by bark beetle. Okay, similarly, causal organism of Dutch arm disease, Ceratostomela almi enters through the wounds caused by bark beetle. Okay, because of the action of this bark beetle, wounds happens on the plant and through that wounds this Ceratostomela almi can infect the plant. Next we are discussing about the entry of pathogens through natural opening. Okay, stomata stomata is a natural opening in plants, right? So through that stomata, this pathogenic organisms can, or these pathogenic agents can enter into the host cell. There is variation in the behavior of germ tube at the time of penetration through the stomata. Okay, there is variation in the behavior of germ tube. Okay, so through it can enter through the natural opening in plants. For example, it can enter through stomata. So there is variation in behavior of germ tube. Okay, so there is a variation in behavior of germ tube at the time of penetration through the stomata. Okay, in Puccinia graminis triticum. In Puccinia graminis triticum, the uridospore. Okay, so this is a spore produced uridospore, which is a spore produces by Puccinia graminis triticum. So this uridospore germinates and forms a germ tube. Okay, so here, so this is a spore, uridospore. This uridospore germinates and forms a germ tube which on approaching stoma. Okay, so here you can see that this is a stoma, right? This is a stoma. Okay, on approaching the germ tube, while approaching towards the stoma, it swells at the tip. Okay, the tip of the germ tube swells to form the episorium. Okay, so in Puccinia graminis triticum, the uridospores germinate and producing germ tube. While they are approaching towards the stomata, it swells at the tip, form the episorium in the stomata aperture. From the episorium, a blade-like wedge grow through the stomatal slits. Okay, so look on to the second one. So here you can see that through the stomatal slit, a blade-like a wedge grows through the stomatal slits and swells inside. Okay, so this blade-like stomatal slits, this blade-like wedges which passes through the stomatal slits and swells inside to form a substomatal vesicle. Okay, so look on to the figure. Here you can see that this is a blade-like wedge which pierces or which passes through the stomatal slits and swells inside. So here it swells, this blade-like wedge which grows through the stomatal slits swell inside to form the substomatal vesicle. Okay, and from which the substomatal vesicle, the hostoria penetrating the cells are produced. Okay, hostoria means what is the function of the hostoria? This act as an absorptive function and it absorbs nutrients and water from the host. Okay, look at the case of Peronospora distractor. Okay, this Peronospora distractor which is infecting onion leaves, the germ tube continues to grow after the formation of first episorium. Okay, so here look on to this. Here the spore, this is a spore. This spore germinate. As a result of germination, it produces spore germ tube. Okay, so here you can see the stomatal aperture there. So once they are approaching towards the stomatal to the stomata, it forms the episorium. Okay, in the cases of Puccinia graminis triticum, from the episorium or through that stomatal aperture, a wedge-like structure which passes through the stomatal slits and swells inside and form a substomatal vesicle, and from that substomatal vesicle the hostoria must formed in cases of Puccinia graminis triticum. But in Peronospora distractor which are infecting onion leaves, the germ tube continues to grow after the formation of first episorium. Okay, so here the germ tube swells here, that is the first episorium. Once the first episorium is formed, and then the germ tube is again continues to grow in Peronospora distractor. Okay, so we have discussed that the entry through the natural opening is varied. is variation. They're having variation. Okay, in Paxinia gramineous tritichy, once they come in contact with the stomata, or they came close to the stomatal opening, aprosoria is formed, and then blade like wedges which passes through the stomatal slits, and inside it swells again and form the substomatal vesicle, and from the substomatal vesicle it forms the haustoria in case of Paxinia gramineous tritichy. And is Pernospora distracta, which is which are infecting onion leaves, the germ tube continues to grow after the formation of first aprosoria. Okay, so here look onto the figure, you can see that this is the formation of first aprosoria, and after the formation of first aprosoria, this is not forming the blade like wedges. This germ tube again continues to grow. Okay, and in pseudo Pernospora cubensis, the hyphae penetrate the stomatal aperture and swells to form substomatal vesicle, from which in turn other hyphae grow to form haustoria in the adjacent cells of the leaves. Okay, so this is the pseudo Pernospora cubensis. So here the hyphae penetrate the stomatal aperture and swell to form a substomatal vesicle, from in turn which hyphae grow to form haustoria into the adjacent cells of the leaves. And in Mycophylla musicola forms a small structure called stomatopodium. Okay, so here it forms a small structure called stomatopodium. It forms a small structure called stomatopodium. It forms a small structure called stomatopodium over the pore of the stoma. Okay, so you can see that this is the pore of the stoma. Over the pore of the stoma, it forms a small structure called stomatopodium over the pore of the stoma after growing for few days on the surface of leaf. Okay. So now we are discussing about the entry of pathogens through natural openings. Okay, we have covered the stomata. Next is the lenticels. Okay, Sclerotinia fruticola, brown rot of fruits, Streptomyces cabecis, scab of potato, Phytophthora araceae, Macaulay disease of wheat. Okay, so these diseases are caused by the entry of pathogen through lenticels. Okay, so you all know what is lenticel. Okay, what is lenticels? These lenticels, which are the pores of a woody plant, pores in the stem of a woody plant that allow gas exchange or gaseous exchange between the atmosphere and the internal tissues. Okay, lenticels, which are the pores in the stem of woody plant, and what is the function is that it helps in the gaseous exchange between the environment and the internal tissues. Okay, so this is the natural pore in the stem, and through that natural pore, this pathogenic organisms or these pathogenic agents or this pathogen which are entering through this lenticels and cause infection on that particular plant. Okay, next is hydatoids, Xanthomonas campestris, which cause black rot of crucifers. Okay, so you all know what is hydatoids. Okay, hydatoids means is a pore that exudate water on the surface of leaf of the margin. Okay, so hydatoids means it's a pore that exudates water. It's a pore that exudates water on the surface or margin of leaf of higher plants. Okay, in higher angiospermic plants, on the margin of the leaves, it having a pore that helps in the exudation of water, and that pore which are seen on the surface of the leaf of higher plants, that is responsible for the secretion of water into the external environment. Okay. So, through hydatoids in higher plants, through hydatoids, it's a natural pore in higher plants, or in angiospermic plants, these hydatoids are the natural opening which are present on the surface or leaf margin of higher plants or angiospermic plants. Okay, through this pores present on the leaf margins, this pathogen Xanthomonas campestris, which enters through this hydatoids and cause diseases on that particular plant. Okay. Direct penetration. Most fungi, nematode and parasitic higher plants. Okay, most fungi, not all fungi, most fungi, nematodes and parasitic higher plants are capable of penetrating the host surface directly. Okay, direct penetration. Okay, what do you mean by direct penetration? They does not require external agents for penetrating into the host cells or tissues. For penetrating into the host cells or tissues, they do not require any external agents. Okay, so most fungi, nematodes and parasitic higher plants are capable of penetrating the host surface directly. However, the plants are provided with different mechanisms of defense, which include structural features of the host, presence of chemical coverings on the cell walls, and anti-infection biochemical nature of the protoplasm. Okay, so many nematodes, fungi and parasitic higher plants that does not require external agents for penetrating into the host cells or tissues. Okay, so they penetrating directly into the host surface. Okay, they directly penetrating into the host surface. They do not require any external agents. Okay, so however the plants, okay, as we all know that our system, okay, while we are taking human as an example, when a foreign organism or when a foreign pathogen, foreign body which is entering into our body, what happens? Our immune system will recognize it and act against that foreign body, right? The same happens on the plant also. Okay, so once the pathogen enters into the host cell, the defense mechanism of the plant will activate and they act against it. Okay, so the plants are provided with different mechanisms of defense. We will discuss about the defense mechanism of plant in our later classes. On the plant pathology, we have to discuss about the defense mechanism of plants. Okay, there is a question also there. While we are discussing previous question, we have seen that. Okay, there is a question regarding the mechanism of defense mechanism in plants. Okay, we will discuss that in detail in our coming classes. So here the plants which having different mechanisms, which include structural features. Okay, the different mechanism means it having a structural features of the host. The defense mechanism is, it can either be a structural feature of the host, or the presence of chemical coverings of the cell wall, or the anti-infection biochemicals which are present on the protoplasm. Okay, so anti-infection biochemical means if that are present on the protoplasm, if an infections agent may entering into the protoplasm, that anti-infection biochemical or that biochemical which act against this pathogen, and it can kill those pathogen. Okay, hence the pathogen should have mechanisms to overcome. Okay, so there are natural mechanism in plants. Okay, if there is natural mechanism in plant, and after that If there is natural mechanisms, the infections are still being there in plants. Okay, so that means these pathogens will overcome the defense mechanism, or it will overcome these barriers for direct penetration. Okay, there are certain defense mechanism in plants, okay, to make infection or for penetrating into the host cells. This fungi or nematodes or this parasitic higher parasitic higher plants which are capable of overcoming the barriers. Okay, so the pathogen should have mechanism to overcome these barriers for direct penetration. Okay, for direct penetration, this pathogens should overcome these barriers for direct penetration. Then only it can cause infection on that particular plant. Okay, so what happens if it cannot overcome the barrier? The infection agent will die there by the natural defense mechanism of the plant. Okay, so here to carry out the infection process in a successful manner, these pathogens which are able to overcome these barriers. Okay, breakdown of physical barrier. Okay, viruses have no physical force or enzyme system. Okay, so viruses does not have any physical force or enzyme system of their own to overcome structural or chemical barriers of the host. Okay, so in the case of viruses, that viruses do not have a force, not have a physical force or enzyme system of their own to overcome structural or chemical barrier. Okay, we have discussed that this viruses, plant viruses which may cause infection on plants only through the wounds. Okay, if a wound happens on the plant, through that wound this can infect the plant. Otherwise, they do not have any specialized structures for penetrating into the host cell, and they mainly enter into the host plant through the wounds happens on the plant. Okay, therefore come in contact with the host protoplasm only through wounds. Okay, so the only possibility of viruses being there in the plant is that it may come in contact with the protoplasm through the wounds happen on the plant. Okay, bacteria are mostly weak parasites. Okay, bacteria are mostly weak parasites, cannot employ force to effect a penetration. Okay, bacteria are mostly weak parasites and cannot employ force to effect penetration. Fungi and nematodes are the only group of plant pathogens. Okay, fungi and nematodes are the only group of plant pathogens that employ force for direct penetration of the host. That point we have already discussed that. Okay, so this fungi and nematodes. Okay, these fungi and nematodes are the only group of plant pathogens that employ force for direct penetration. This bacteria or viruses cannot enter into, or these viroids or these molecules cannot directly enter into the host plant. Okay, mainly they are entering into the host plant through the insect puncture or through insect punctures or through the wounds happen on the plant. Only through that they can enter into the host plant. And this fungi and nematode are the only group of plant pathogens that employ force for direct penetration. Fungi penetrate host plants directly through a fine hypha produced directly by the spore or mycelium or through a penetration peck produced by an episorium. Okay, so fungi penetrate the host plants through a fine hypha. Okay, this fine hypha which is produced either by a spore or mycelia or through a penetration peck produced by an episorium. Okay, so we have discussed that on the penetration peck, the swelling on the germ tube is the episorium, and then in cases of Puccinia gramineous triticum, a blade like wedges which are passing through the stomatal slits, and inside it forms a substomatal vesicle, and then it forms a hoostoria. Okay, so fungi penetrate the host plants directly through a fine hypha produced directly by the spore or mycelium or through a penetration plug produced by an aprosoria. These structures exert pressure. Okay, this hyphae or this mycelium which exerts pressure on the surface. Okay, so if this is the host plant surface and here hyphae which are formed on the host plant surface, right? Okay, so that these structures, this germ tube, this penetration plug, aprosoria, hostoria, these structures will exert pressure on the surface. Okay, so it exert pressure on the surface. That means here the epidermis will stretch. Okay, when we are exert pressuring on the surface, it will cause the stretching of the epidermis. Okay, what happens if the epidermis is being stretched? That part will become thin. Okay, so as a result of this pressure exerted by this structure, this epidermis is stretched. Okay, the stretched part of the epidermis is thin, and through that region they can penetrate into the inner cells of the plant. Okay, so become thin, then the infection peg punctures and it affects its entry. Okay, so if the epidermis is stretched, that part will become thin, and that penetration peg which can pierces into that thin region and it can enters inside the host plant, and so that it can come in contact with the host cells or tissues, and they are capable of infecting that host cells, and in case of the fungi they can absorb their nutrients and sources, their mineral sources and nutrients they can absorb from the host cells through the hostoria. Okay, so they get the nutritions also there. Okay, and if they are getting the nutrition, they will continue their growth and development. Okay, breakdown of chemical barriers. The host is provided with defense mechanism against inversion, which include presence of cuticular layer on the epidermis. Okay, breakdown of chemical barriers. Okay, the host is provided with defense mechanism against inversion, which include presence of cuticular layer on the epidermis. Okay, so as we all know, if this is the epidermis, on top of the epidermis it have the cuticle. Okay, it having a cuticle. Okay, presence of cuticular layer on the epidermis. The host is provided with defense mechanism against inversion, which include, okay, so naturally the plants which having the defense mechanism, that means on the epidermis it having a thick cuticle. Okay, so naturally the foreign invaders cannot enter into the epidermis. Okay, lack of suitable nutrients for the pathogen in the host cell. Lack of suitable nutrients for the pathogen in the host cell. Presence of inhibitory or toxic substances in the host cell. Exudation of substances toxic to pathogen or stimulatory to antagonists of the pathogen. Okay, mainly this plants having the defense mechanism. Okay, so example, the glands in leaf hairs of Bengal gram contain malic acid, which is antifungal and provide resistance to infection by the rust fungus. Okay, so this rust fungus are the Uromycerius erytini. Okay, so here naturally the plants having this defense mechanism against the inversion. Okay, so the glands in leaf hairs of Bengal gram contain malic acid, which is antifungal and provide resistance to infection by rust fungus, Uromycerius erytini. Okay, similarly protocatechuic acid and catechol in the red scales of onion provide resistance to onion smut pathogens. Okay, Coleotrichum cercinans. Okay, so similarly protocatechuic acid and catechol in the red scales of onion provide resistance to onion smut pathogen. Okay, to overcome this physical and chemical barriers, okay, there are natural defense mechanism in plants. Okay, to carry out the infection in
plant, this pathogen should overcome this barriers. Okay, there are physical and chemical barriers are there. So to infecting the plant, they have to overcome this chemical and physical barrier. So how they overcome this? How the pathogen overcome this physical and chemical barrier? Okay, so to overcome this physical and chemical barriers, the fungi produce various enzymes. Okay, so to overcome this physical and chemical barriers, this fungi which are capable of producing various enzymes, toxins, organic acid and growth regulators. Okay, so this we have discussed in detail in the plant defense mechanism. Okay, for the time being, just understand that to overcome this physical and chemical barriers, this fungi produce certain enzymes or various enzymes, toxic organic acids and growth regulators. Okay, by the production of this growth regulator, organic acid and toxic toxic organic acids and enzymes, this can overcome the physical and chemical barriers of the plant defense mechanism. Okay, so next we are discussing about the entry through non-cutinized and cutinized surfaces. Okay, so seedlings, root hairs, buds, flowers, leaves, nectaries, these are the non-cutinized surfaces. Okay, so the pathogen can enter through this non-cutinized surfaces. Okay, seedlings, downy mildew of jowar and bajra, Sclerospora graminicola, weech bun disease, Tilletia caries and Tilletia foetida. Okay, so these diseases are caused by the infection of this particular pathogen and they are entering into the plant through seedlings. Okay, and root hairs, wilt causing fungi, Fusarium species, club rot of cabbage, Plasmodiphora brassicae, red rot of cotton, Phaematotrychum omnivorum. Okay, so these diseases are caused by the entry of pathogen through root hairs. Buds, Pyrrhus fungi, Euromyces species, witch's broom of cherries, Tavrina cerasii. Okay, so these diseases which are caused through the entry through buds. And flowers, loose smut of wheat, Usilaga nuda triticii, ergot of fry, Claviceps purpura. Okay, so these diseases are caused by the entry of pathogen through flowers. Okay, leaves, basidius spores of white pine blister rust of fungus, Cronarium ribicola, germinate and grow down into branches and leaves where Ascia are produced. Okay, what do you mean by Ascia? Ascia are the cup-like structures of some rust fungi that contains a chain of Asciospores. Okay, Asciospore is a cup-like structure which contain a chain of Asciospores. Okay, these Asciospores which are seen in some rust fungi. Okay, we will discuss that in detail under cryptogammas in unit 2. We need to discuss about fungi in detail, so that time we will discuss about the Asciospores in detail. Okay, so for the time being, just to know that this Asciospore, which is a cup-like structure of some rust fungi which forms a chain of Acidiospores. Okay, and next is nectaries, fire blight of apple, Erwinia amylovora. Okay, so this fire blight of apple disease which is caused by the entry of pathogen to nectaries. Okay, and cuticle. Okay, so we are discussing about the entry through non-cutinized and cutinized surfaces. Okay, cuticle, leaf spot of spinach, Cercospora beticola, early blight of solanaceous plants, Alternaria solani, Tika disease of groundnut, Cercospora personata. Okay, these diseases are caused by the entry of pathogen through the cuticle. Okay, under infection process we have discussed, so far we have discussed the pre-penetration and penetration. Next we are moving on to the post penetration. Okay, so inversion and colonization. Okay, post penetration means inversion and colonization. Infection is the process by which pathogens establish contact with the susceptible cells or tissues of the host and derive nutrients from them. Okay. So, inversion and colonization. So, what do you mean by inversion? Okay, inversion means, foreign inversion means that foreigners may come to a particular region and they take control over that region. Okay, that is what we call as inversion. Okay. So, here inversion and colonization. So, after the penetration, what happens? The pathogen invert into the host cells and they take the control and they colonize inside the host plant. Okay, infection is the process by which pathogens establish contact with the susceptible cells or tissues of the host and derive nutrients from them. Inversion means they take control over the host cell. That means in case of fungi, they are parasitic in nature, or they are parasitic in nature. So, they absorb the food from the living cells. Okay. So, susceptible cells or tissues, they establish once the pathogen infection is the process by which pathogen establish contact with the susceptible cells and tissues of the host and derive nutrients from them. Okay. A parasitic relationship is found between the host cytoplasm and a parasite cytoplasm. Okay. So, after the successful infection, the parasitic relationship is found between the host cytoplasm. A parasitic relationship is found between the host cytoplasm and the parasite cytoplasm. Okay. During infection, pathogens grow and multiply within the plant tissues. Okay. During the infection process, okay, after the successful penetration, this pathogens will grow and multiply within the plant tissues. Okay. They grow and multiply within the plant tissues, so they grow and multiply within the plant cells. Inversion of plant tissues by the pathogen and growth and reproduction of the pathogen. Okay, growth and reproduction of the pathogen is called as the colonization. Are two concurrent stages of disease development. Okay, so what are the two concurrent stages of disease development? One is the inversion of plant tissues. Okay, inversion of plant tissues by the pathogen and growth and reproduction of the pathogen. That is what we call as a colonization. Okay, colonization and inversion. Colonization and inversion both are the concurrent stages of disease development. Okay, what are the two concurrent stages of disease development, which are inversion and colonization. Okay, inversion means they take the control over the cells and colonization means growth and development of the pathogens inside the plant cells or tissues. In ectoparasites, the main body of the pathogen lies on the surface of the host. Okay, so ectoparasite. Okay, if this is the surface of the plant, okay, the main body of the pathogen lies on the surface. So, if we are considering this as the main body of the surface of the pathogen, this is lies on the surface of the host with only feeding organism. Okay, feeding organism means hostoria is a feeding organism or the absorptive structure. They pierces into the, or they penetrating into the tissues and take nutrients and their requirements for the growth and development of the ectoparasites main body. Okay, so ectoparasite means the main body is on the main body of the pathogen which lies on the surface of the host with only feeding organisms like hostoria will pierces into the tissue and they take up the nutrients from the tissues. Okay. Example, most of the powdery mildew which are ectoparasitic in nature. Okay, some fungal parasites developed both external and internal mycelium. Okay, some fungal parasites which may develop both external and internal mycelium. Example, Rhizotonia zolani. Rhizotonia zolani, which develops both external and internal mycelium. The endophytic parasites or endoparasites grow subcuticularly. Okay, so ectoparasites and endoparasites. Ectoparasite means the main body which is on the surface of the host. Okay, endophytic parasites means endoparasites or grow subcuticularly, okay, below the cuticular level, and diplocarpon rosae, which cause black spot of rose in parenchymatous tissue. Okay, some pathogens are endobiotic, that is mycelium is not produced, and thallus means the plant body, sorry, thallus means the body, okay, not the plant body. Thallus means to mentioning the lower forms, okay, in algae, bryophytes and fungi we are using the terms thallus instead of plant body, okay. So some pathogens are endobiotic, that is mycelium is not produced and the thallus is entirely present within a host cell. Example, Syncytium endobioticum, okay. Bacteria invade tissues intercellularly but also grow intracellularly when parts of cells dissolve, okay. Bacteria usually invade tissues, intercellularly but also grow intracellularly when parts of the cell walls dissolve, okay. So no need to explain those things. Viruses, viroids, molecules and fastidious bacteria, okay. Viruses, viroids, molecules and fastidious bacteria invade tissues, okay. What do you mean by fastidious bacteria? We have discussed that term, okay. It requires specialized condition and substances, okay. It requires specialized conditions and substances for their growth and development, okay. Molecules, the other term for molecules is mycoplasma, and viruses, viroids, molecules and fastidious bacteria invade tissues by moving from cell to cell intracellularly. Infection caused by microbes may be local, okay. Involve single cells or few cells or small area. Infection caused by microbes may be local. The infection caused by microbes may be local. That means that infection happens only to a single cells or only to a small region. Or systematic, pathogen spread and inwards, okay. Or in one case that it is the microbes may be local, that means it involved with single cells or a few cells or to a fewer region only, okay. And in certain other cases where pathogen spreads and inwards most of all susceptible cells and tissues throughout the plant, okay. On the other hand, in some cases the pathogen may be localized, it is few pathogen is involved with a single cell or to a small region. On the other hand, certain pathogens which spread all over the plant cells and tissues, okay. Sclerospora graminicola, okay. The time interval between the inoculum and appearance of disease symptoms is called as the incubation period, okay. So we have discussed that inoculum and we have discussed the term there, okay. The time interval between inoculum and appearance of disease symptoms is called the incubation period, okay. So what do you mean by the incubation period? The incubation period is the time period between inoculum and appearance of disease symptoms, okay. The time period between inoculum and the appearance of disease symptoms is what we called as the incubation period. Exit of the pathogen. After inversion and colonization of the host, the pathogens come out of the host to maintain the continuity of the infection chain or disease cycle and escape death due to overcrowding, okay. So after inversion and colonization, the pathogen may come out from the host to maintain the continuity of infection cycle or disease cycle and escape death due to overcrowding, okay. So by the exit of this pathogen from the host, it help them to escape from overcrowding and it can also escape their death, okay. Overcrowding means it can cause death, okay. So to avoid that, it will exit the pathogen, okay. After inversion and colonization of the host, the pathogen may come out of the host for maintain the continuity of the infection chain. Once the pathogen exit from the host, okay, once the pathogen exit from the host, they survive and are disseminated to other hosts and continue the infection cycle. Okay, once the pathogen may exit the host, they survive. Okay, once the pathogen exit the host, they survive and are disseminated to other hosts and maintain the infection cycle. Okay, viruses can exit only with the living protoplasm. Okay, so we all know that viruses which can multiply, which can grow, multiply inside the living cells only. Okay, viruses can exit only with the living protoplasm and hence disseminated through their vectors like insects, fungi and nematodes. Okay, viruses, for their survival, virus needs a living cell. Okay, so the dissemination of these viruses which are disseminated through vectors like insect, fungi and nematodes. In case of bacteria, the bacteria ooze out in the form of slime on the host surface. Okay, so if this is a host surface, they come out as a form of slime on the host surface, from where they can be disseminated through water and insects. Okay, they can come out in the cases of bacteria, they can come out in the form of a slime like structure and on the host surface. Okay, they form a slime like structure. After the successful inversion and colonization, they may exit the host. Okay, so during the exit, they form slimes on the surface of the host and through with water and insects, these are disseminated. Okay, these slimes which are disseminated through water and insect. Okay, however, the fungi have most elaborate system. Okay, however, the fungi have most elaborate system of exit. Most plant pathogenic fungi grow out on the host surface and produce repeating spores. Secondary inoculum, usually asexually under favorable conditions. Okay, however, the fungi have most elaborate system of exit. Okay, the fungi having the most elaborated system of exit. Most plant pathogenic fungi grow out of the host surface. Most plant pathogenic fungi grow out of the host surface and producing repeating spores. Okay, so these are the secondary inoculum. This inoculum which are capable of causing infection, usually asexual under favorable conditions. Okay, so asexual spores which are produced under this fungi, which are produced asexual spores under favorable condition. Okay, the spores thus formed are disseminated through. Okay, the spores thus formed are disseminated. The spores thus formed by the fungi which are disseminated by which are disseminated through wind, water, soil, seed, vegetative propagating material, agriculture implements, etc. Okay, so here the fungi which have the most elaborate system of exit. So they produce the spores essentially and under unfavorable conditions. Okay, so this most plant pathogenic fungi grow out of the host surface and produce repeating spores called the secondary inoculum, and usually asexually. Asexual means there is no chematic fusion, right? And this asexual spores which are produced under favorable condition, and these spores thus formed are disseminated through wind, water, soil, seed, vegetative propagating material and agriculture implements. Okay, so that's all about the phenomenon of infection and infection process. Hope it's all clear for you. Okay, for the more videos related on Botany Optional, follow our channel. Thank you.